lecture, I've put together this short video to provide you with some background information on genetic mapping so that when we get to class, we can have more time for your questions and to actually practice a few problems. So a genetic map is simply a representation of the order and distance of genes along a chromosome. So at its simplest level, this is a genetic map. A, then B, and then we'd also provide an estimate of the distance between them, say 33.4 centimorgans. We'll talk more about what a centimorgan is later, but for now, know that this is a genetic map at its simplest. So key to estimating this order and this distance is a concept called recombination. And recombination is the generation of new combinations of traits and alleles due to crossing over between homologous chromosomes during meiosis. So I can illustrate that here. We have two X's representing our two chromosomes. So diploid organisms have, like humans, have two copies of each chromosome, one from their mother, one from their father. And these chromosomes are where our genes are and where our alleles are. So this individual is heterozygous, big A, and big B, little b. So he's heterozygous for two traits, A and B. And on the first chromosome, he has his two dominant alleles, big A and big B. And since the chromosomes have duplicated prior to meiosis, we have two arms for those chromosomes. And then on the other chromosome are his recessive alleles in this case, little a and little b. And again, they're duplicated. Now, during crossing over, segments of DNA between these chromosomes are physically exchanged. So this arm would go over here to that chromosome, and this black arm would go over to the red chromosome. The result of that crossing over produces new chromosomes with new combinations of alleles. So at the end of crossing over, CO for short, we have these outer arms of the chromosomes that didn't undergo any crossing over. They look the same. Big A, big B, little b. But for the arms that did undergo crossing over, you'll notice some differences. First, the red arm is over here, the black arm is over here, which means the alleles have moved too. We have little a, big B, which is a new combination, and big A, little b. Once meiosis is complete and these sister chromatids have been pulled apart into sister chromosomes to form new chromosomes and packaged into gametes, we have these outer arms that didn't undergo any crossing over. They look the same as the parent's original chromosomes, big A, big B, and little a, little b. And in the middle we have these new combinations, little a, big B, and big A, little b. These are new combinations of alleles not seen in the parents, so we call these recombinants. Now, how frequently recombination between two genes like A and B occurs depends on how far apart they are. If they're very far apart, say opposite ends of the same chromosome like we have here, then recombination will happen very frequently. If they're close together, say right next to each other, then recombination will happen a lot less often. In that way, we can use measures of recombination frequency as an estimate of the genetic distance between two traits, A and B. So I'll erase this board, and when we come back, I'll show you how we actually measure recombination frequency and estimate that distance. So we're back. I've taken the chromosomes that we generated from the last board from our heterozygous dominant individual, the one that was big A, big B, little b, and I've shown those here in black. We have the two chromosomes that were on the outside that look exactly like the parent's original chromosomes, and we have in black here those two recombinant chromosomes, and I've outlined these in red. You'll see I've also combined them or mated these chromosomes with chromosomes from another individual, one that is homozygous recessive, little a, little b, and I've shown those here in red. So what I've done is I've performed a genetic cross when we take a heterozygous dominant individual and mate them with an individual that is homozygous recessive. We call this a test cross, and this is what we use to estimate the frequency of recombination between two or more genes. So in this case, from our test cross, we've produced a bunch of progeny or offspring or children. I have 100 progeny that look exactly like our heterozygous dominant individual. They have big A, big B, little b chromosomes. I have another 100 offspring that look exactly like our homozygous recessive individual, little a, little b, little a, little b. And then I have several progeny that have inherited one of these recombinant chromosomes from the last board. 50 that have the little a, big B. Their total genotype is little a, big B, little b. And another 50 that inherited the other recombinant chromosome, big A, little b, little b. For a total of 300 progeny altogether. Now to estimate the frequency of recombination between A and B, we simply add up the number of recombinant progeny and divide them by the total number of progeny, and then multiply that number by 100 to get a percentage. So if we wanted to estimate the recombination frequency between A and B, shown here, we would take the number of recombinant progeny. In that case, it's 50 plus 50, and divide that by the total number of progeny, in this case, 300. We'd multiply that final number by 100, and you'd see we'd get 33.4 centimorgans. And so then we can fill that in our map. A to B is 33.4 centimorgans. Now, a centimorgan is the unit of genetic distance. 1 centimorgan equals 1% recombination, or 1% recombination frequency. Sometimes this is also called 1 map unit. The centimorgan was named after Thomas Hunt Morgan, a famous fly geneticist, and it was named by one of his undergraduate students. This student, while still an undergraduate, did exactly what we've done on the board. He produced a test cross for 2 traits, estimated the frequency of recombination between those traits,